Hello, my AS students. Today we will be explaining the last activity in our chapter Biotechnology and Immunology. We are going to talk about monoclonal antibodies. How are these antibodies experimentally produced? First, as an introduction, what are antibodies and what are the cells of the body that produce the antibodies and in which situations are these antibodies produced? The introduction of a foreign substance called antigen into the body provokes or triggers an immune response that is characterized by the production of antibodies by immune cells called B lymphocytes. Here we have a B lymphocyte expressing on its surface antibodies. Once the B lymphocyte is activated by a certain type of antigen, the B lymphocyte transforms into a plasmocyte, and the plasmocyte, which is the activated B lymphocyte, releases several amount or a high quantity of antibodies which are specific for the same type of antigen or for the introduced antigen. So we say that each type or clone group of B lymphocytes produces only one type of antibodies that are specific for one type of antigen, more specifically for a part of the antigen called epitope. So the antibodies produced by the same type of B lymphocytes are called monoclonal antibodies. Mono means one, clonal group, antibodies. Here we have an antigen expressing different epitopes. So the antibody, the Y-shaped molecule here, the antibody, will bind by complementarity to a part of the antigen called epitope. So each antibody is specific for an epitope for a part of the antigen, and all antibodies produced by the same type of B lymphocyte are called monoclonal antibodies. Many types or clones of B lymphocytes produce many types of antibodies. Thus, the antibodies produced by different B lymphocytes are called polyclonal antibodies. Poly means many or several, clonal means clone. So, many groups of B lymphocytes produce many types of antibodies. Here, as you can see in this document, we have different types of antibodies produced by different types of B lymphocytes. That's why they are called polyclonal antibodies. Remember that when you see the exclamation mark, this means that this slide and this information should be studied, even memorized. Now, the question is, are the antibodies produced in our body monoclonal or polyclonal? The answer is the antibodies produced in our body are polyclonal. Why? Since our body possesses a variety, a repertoire of B lymphocytes produced following the natural infections we encounter during our life. Thus, we are immunized, we have an immunity, an immunological memory against one or more diseases or pathogens depending on the type of the pathogen. So our body develops a variety of immune responses against a wide uh, group of pathogens. That's why the antibodies produced in our body are not only monoclonal, but they are polyclonal. So in the next video, we are going to see how are monoclonal antibodies experimentally produced. Since, as I have explained, our body does not contain monoclonal antibodies but polyclonal. So how can we obtain monoclonal antibodies in the lab? All this will be discussed in the next video. Thanks for listening.